Okay, so today we're going to be talking about topic 6.5, which is all about the neuronal system. So we know that we have the nervous system, whose main job is to take in information about our environment and coordinate some kind of response with your body to do that. And we know that the main center is the brain, which is the central nervous system, and that's going to go down your spinal cord and then branch off into the different peripheral nervous system parts from there. So the main cell type in the nervous system is the neuron, right? And so a neuron's job is to receive signals from another neuron and then send that signal down to either another neuron or whatever target tissue it's going to impact. So we call these electric signals impulses, which generate something called an action potential. And if you're feeling there like a little stressed out already because I'm throwing all these new words on there, just appreciate that you're basically a meat sandwich that's sitting there trying to figure out how it works. And so there's going to be a lot of new vocabulary. It will all make sense in the end, right? Okay, so let's look at so the cell, the neuron, main job, receive and transmit signals, right? And so let's take a look at the neuronal structure. So you only find neurons in animal cells, right? So this is just your standard animal cell. So a neuron, like every other animal cell, is going to have a nucleus, it's going to have a cell membrane, it's going to have mitochondria to generate ATP, it's going to have a Golgi apparatus and vesicles all over the place. So it will have a main part, right? And so the main part, the main cell body, is something called a soma. And you'll notice that compared to other cells that we've seen, the neuron structure is really different. So we have, again, remember, its main job is to receive and send signals, right? So it's going to receive signals on one end and transmit them down something called an axon, which is this long branching structure right here. And so we have on the left side the dendrites, which are going to receive a signal from another cell, and signals will get transmitted down an axon through the axon terminal, which are going to then connect to a different neuron. So no neuron does its job by itself. It's usually these huge networks of possibly millions of neurons, and so they're transmitting these signals along the way. So of course IB likes to focus on really esoteric structures within the neuron, right? So most of the action that we're going to concern ourselves with happens in the axon itself. We're kind of going to ignore the soma and the dendrites for right now and focus primarily on what's going on in these substructures in the axon. So the axon, you'll notice, is covered in these other cells. And so this coverage is something called a myelin sheath, which is produced by a different cell type called Schwann cells. So they're secreting this really lipid-heavy sheath, which is going to help facilitate this transmission of signals. And you'll notice that there's these gaps in this myelin sheath. And so these are called nodes of Raynier, which are going to be important in something called saltatory conduction. And so I just wanted to give you the landmarks of what a neuron looks like before we start talking about what these different parts actually do. So all of these are within the axon. We have a myelin sheath which is covering the outer layer of the axon. And that myelin sheath is produced by Schwann cells. And then these little gaps are called nodes of Raynier that are going to be involved in a mysterious process called saltatory conduction. Okay, so let's look at this myelin sheath, right? So remember that the nerves are conducting signals over really long distances very quickly. And these are electric signals. And without some kind of intervention, that electric signal would decrease over a long distance. It would take much longer for that to happen. So what is happening is these Schwann cells are producing this myelin sheath, which is this structure which is surrounding the actual axon of the neuron cell. And this myelin sheath is going to facilitate that saltatory conduction. And so your neurons, they're generating electric impulses. And these electric impulses, rather than just going straight through the length of the cell, they're actually able to make jumps between these nodes of Raynier. And so that skipping action has a special name, and that is called saltatory conduction. So when I'm talking about that, I'm talking about the electricity really jumping over through these nodes of Raynier. So this is facilitated by this myelin sheath, which is again made mostly of lipids. And so what this is actually able to do is really speed up the speed of the signal as fast as like 100 meters per second, which if you're talking about a cell is really quite remarkable. So we have special names for this electric signal that is being generated, and we call them an action potential, which we can look at in a graph. Like I could, if I wanted to, hook you up to an oscilloscope and see the action potential, see the electron signal coming from your neurons. So these are measuring how the change in voltage across the membrane over time. And so cells are pretty small, so this is going to be measured in millivolts, and it's going to be measured over time. So this is looking at the potential, so difference in electric voltage across your membrane over time. And so this is happening, right now in your brain. As you're even listening and processing this, your brain is generating electric impulses to interpret the words that I'm saying. It's communicating to neurons to try to make sense of the images that you're seeing. So even sitting here right now, you are generating action potentials. And so when I graph this action potential, this is what they look like. That on the y-axis, we have the membrane potential in millivolts, and then the x-axis, we have this how the action potential is changing over time. So when a neuron is at rest, the resting potential is at negative 70 millivolts. And we're going to talk about exactly what is happening in the cell during each of these different changes. But in its normal state, when a neuron is just sitting there, it's sitting happily at negative 70 millivolts. When a neuron fires, it does something we call depolarizing. And I'll explain more about that, what that means. And then as it needs to reset, because, hey, I sent the signal, now I need to reset myself, it goes through a repolarization, and then it usually overshoots it in something called hyperpolarization before it returns to that resting potential. So this is what an action potential graph looks like. So you might have to draw these. You might also, they might point to different points of the graph and ask you like which stage is it in? Is it depolarization? Is it repolarization? And then just to be extra mean, they're going to ask you about the open or closed state of like the sodium channels that we're going to talk about in a second. So yeah, be able to interpret what's happening at these different points in the action potential. So overall, transport in axons and the overall action potential has 3 stages. The first step is to generate that resting membrane potential, which I'm going to tell you up front involves our old friend, the sodium-potassium protein pump. After the neuron depolarizes, we're going to have facilitated diffusion of the different channels. So these are different, right? So facilitated diffusion is the passive transport of both sodium and potassium channels, which is going to depolarize the membrane. And then when that neuron needs to reset, it's going to repolarize, which again involves facilitated diffusion, as well as a voltage-gated potassium channel. So that's going to stop the firing of the neuron, and the whole process will repeat itself. So this is like a great paper 2 question, like explain the movement of ions during an action potential. And you again will be asked to match what's happening in these different steps to what's happening in the action potential graph. So this is just a friendly reminder that protein pumps, we saw these last year. This is a form of active transport where you have some kind of an embedded membrane protein, and its whole job is to move materials from a low to a high concentration gradient. So we're going against our concentration gradient. So because of that, it is energy using, because this is active transport. And the most common example that we've seen so far is the sodium-potassium pump. We saw that during glucose transport in the digestive system, and we're seeing it again here in the neuronal system. So just to kind of review that the steps in the sodium-potassium pump, functionally what's happening is that 3 sodiums are binding. In this case, they are being transported out of the cell. You can flip them any direction that you want. So in this case, in the neuron, it's 3 sodiums going out, and then it's going to bring 2 sodiums in that are going to bind and then bring it inside the cell. So this is flipped. It's reversed from how things were happening in the digestive system where we were transporting sodium out of the cell. In this case, it's still transporting them out. So what this is going to do is you're going to end up with a higher concentration of sodium outside the cell. and a higher concentration of potassium inside the cell. And the long and short of it is that this is the process of which generating that resting potential of negative 70 millivolts. And so you might be asking, okay, why is it negative? You know, so if we look at this in a more general sense, right, this is probably going to make sense. Like here we have our membrane, here we have our sodium-potassium protein pump, which is kicking out 3 sodiums for every 2 sodiums that it's bringing in. And so if you look at this in just a general sense, which side is more positive? Like which side am I putting more positive charges on? It's gonna look like the outside, right? And this actually is what happens. That because I'm, transporting 3 sodiums out for every 2 sodiums or potassiums in, you end up with a net charge of positive on the outside and a net charge of negative on the inside. And so this is functionally what you're following when you're looking at the membrane potential. So this is why you start with a negative 70 millivolts membrane potential in the resting. And these charges are going to flip as these charged ions start moving across the membrane. So in the resting membrane potential, we're at the negative 70 millivolts, where it's really just the sodium-potassium protein pump acting. That's putting those charged sodium ions outside, sodium ions outside, and only the 2 on the inside. So we're more positive outside than inside, and we have this resting potential of negative 70 millivolts. Now, when you depolarize, right, you'll notice that we have a pretty long axon. And so what is really happening is that the signal doesn't go directly across. What it's happening is that you have the closing or opening of different channels along the length of the neuron. And so this is a way to transmit the signal without actually having to run the whole length of it, in that they kind of fire like a row of dominoes, like what you're seeing in this action here, right? That I have one part of the axon that's starting to fire, and it's going to tell its neighbors to pass the signal on. Like I could have you guys close your eyes, and you could tap the person next to you on the right shoulder, and you could transmit the signal just as well. So in this situation, we start where we have more sodiums outside, fewer potassiums inside, with that positive outside, negative inside. So when it depolarizes, what's going to happen at rest, right? The channels are closed, but once the neuron fires, once it depolarizes, right, the signal is going to come through, and it's going to cause these channels to open. And at this point, everybody can run free. They can switch sides, and that is exactly what happens because they're going down their concentration gradients. These are just facilitated diffusion channels. And then what's going to happen is the membrane depolarizes and actually the charges flip. So that's why you see in the action potential graph that we go into positive territory there for a bit. And so once it depolarizes in one area, the signal is able to go down to the rest of the axon. And so now, once that's done, it has to stop and reset. So how that is done is with voltage gated channels. And they only open when a potential is high, meaning only when it's very positive, right? So that would be after the depolarization, and then it's going to close once you get into negative territory. And so how these gated channels work is exactly like it's shown here, that once it's positive, it will open, allowing these, ions to travel back to where they were. And once it is negative, it's going to close up again. So these facilitated diffusion channels look something like this, or like reverse Pac-Mans, right? And so during depolarization, what's going to happen is they're going to open up, right? So depolarized, I'm more positive on the inside. I'm going to open up because I want to reset. It will allow these potassium channels to go out. After it depolarizes, it's going to shut off, right? Because it doesn't want to permit that to happen anymore. And so this stops the escape of those potassiums, and at the same time you have the work of the sodium-potassium channel, so that resting membrane potential is going to be restored because this is stopping those potassium ions from escaping. And so if we look at the action potential, right, so we have these different stages. We have resting potential here, depolarization, and then repolarization. It briefly goes into negative territory, which is called hyperpolarization. Um, so in the resting state, right, this is where you have the activity of the sodium-potassium pump, which is pushing 3 sodiums out and 2 in. You have that negative 70 millivolts potential, resting membrane potential, and at this point the sodium and voltage and potassium gates are closed. During depolarization, the sodium voltage gating channels are going to open while the potassium remains closed. When we're ready to reset, it flips. That the voltage gated channels for potassium are open and the sodium is closed. So that's what we're going to focus on for right now. We'll pick up synapses next time.